Okay, so this lecture is going to focus only on the cell membrane. In the last chapter, we did an introduction to the cell. We learned all the different parts, what they did, and somewhat about how they work together. In this chapter, we're going to focus specifically on the membrane and the membrane's role in controlling what enters and leaves the cell and how cells communicate with other cells. So we're going to start with a little bit of history. Okay, so in the early 20th century, they knew that the cell membrane contained phospholipids, cholesterol, and proteins. However, they didn't really understand how it was arranged. So if you look at this electron micrograph, you'll see that this is actually two cells side by side, and you can see basically two phospholipid bilayers. They thought, because it looks uninterrupted, that the proteins must fit somewhere on the surfaces, not stuck in the membrane, because it appears to be sort of an uninterrupted bilayer. So as technology improved, they were able to do a technique called freeze fracture. And that's what this is showing here. So what they were able to do is cool the membrane down enough that they could then separate the bilayer. And when they did that, they were then able to see that the proteins actually were stuck in the membrane in different areas, and that's where they got the fluid mosaic model. And what that really refers to, fluid, comes from the fact that the phospholipids are actually liquid-like. They're not solid. And then a mosaic is sort of a picture made of tiles. I always give the example, if you go to Disney World and you walk through the center portion of the castle, on the wall there's a mosaic of the story of Cinderella. You can actually see little tiles making up all of the pictures. So that's what a mosaic is, and that's why it's called the fluid mosaic model. The fluid is referring to the liquidy part, which is our phospholipids, and then the proteins form a mosaic. They're kind of randomly placed tile-like structures in the membrane. Okay, so the fluid mosaic model, first of all, we talk about the phospholipid bilayer. The hydrophilic heads, remember hydrophilic, water-loving, they point outwards towards the outer part, the extracellular matrix, and the second layer then they point in towards the inside of the cell. The tails are hydrophobic, they are water-hating, so they face each other in the bilayer. Also, remember we said it's called the fluid mosaic model, and that's because the phospholipids are unsaturated. Remember, that means they have double bonds, and whenever there's double bonds, that causes these kinks in the membrane, and that's where the fluid part comes from. In fact, if the membrane gets too cool and it starts to solidify, it won't work properly. It won't be able to do the jobs that it normally does. So it's important that it stays fluid. The proteins, there's different types of proteins we're going to talk about in a minute, but some of the proteins are extrinsic, and that means they are actually on the surface, and then others are what are called intrinsic, meaning they're within, and a lot of them are transmembrane, meaning they go completely through the membrane. They stick out on the two sides. And then in animal cells only, we see cholesterol. Cholesterol actually adds to, at cool temperature specifically, it helps to keep the membrane fluid. Alright, and here's a diagram of a protein in the cell membrane, and hopefully you recognize the alpha helixes. Remember, this was from our biochem chapter where we talked about how proteins had secondary structures, and it turns out, if you notice, that the alpha helixes are sort of aligned with the lipid portion, and that would mean that the alpha helixes must be hydrophobic. So the reason they couldn't see the proteins in the original electron micrograph is because if electron micrograph was staining, let's just say the phosphate part, it was staining the hydrophilic portions, then it would also have stained the hydrophilic parts of the protein that were sticking out, and it wouldn't have stained this part. So you wouldn't have been able to see the protein. It would have been invisible. It would have blended. This picture here is showing just a phospholipid bilayer. Notice that the phospholipids, they show them all bent, and this would have to be from an animal cell because here's your cholesterol. Remember, it's a steroid, so you have 4 fused rings, and that would represent the cholesterol in the cell membrane. This is just showing a diagram of different representations of a phospholipid. Back when we were doing biochem, we would have talked about it on this level on the right, where we actually see the phosphate here, and then the 2 fatty acids. Today, and from here on out, we represented just the a phospholipid would just be the hydrophilic phosphates, just a circle, and then the two tails are two sticks that kind of have bends in them. Alright, so this is just another diagram showing the complexity of the cell membrane. And here, this is kind of showing what we mean about the unsaturated lipid tails. If you notice how they would prevent this from becoming a solid. This is what would happen if they were saturated. They would pack together and the membrane would solidify. And then here, this is showing cholesterol stuck in the membrane, which would also help. It says at higher temperatures it's not really helpful. It can actually reduce the fluidity of the membrane, but particularly at lower temperatures, this would be really, really important in animals as far as keeping the membrane fluid. Okay, so the lipids, remember, the phospholipid bilayer is a liquidy layer, and so the phospholipids are not stuck where they are. So for example, if this was a phospholipid that let's say I'm going to color it in, what can happen is this phospholipid can move, and maybe later I find it over here. What would not happen is this phospholipid would not go through to here and then end up on the other side. And the reason why makes sense, because this phosphate is hydrophilic and this whole section here is hydrophobic. So there's no way that this hydrophilic part would be able to pass through here to get to the other side. Alright, and the other thing that we're going to see is that the outside and the inside of the membrane are not going to look the same as each other because they have different jobs. For example, the outside of the membrane, one of the most important things that's going on there is you're going to have receptors to pick up signals from, say, hormones in the blood. You're going to have ID markers that are going to identify this cell as belonging so it doesn't get attacked by white blood cells. Obviously, white blood cells are not going to be inside the cell. They're going to be outside, so your receptors need to be outside, and your ID markers need to be outside. So that makes sense. This next slide that I have was just a little, I guess, self-check. If you had a fish that lived in an Arctic environment, how might its cell membrane differ? We talked about this in class. For example, we know that the more unsaturated fatty acids, the more likely a membrane would stay fluid at lower temperatures. So maybe it would have a higher concentration of fatty acids in the membrane. Another thing is, we said animal cells have cholesterol. And if it's an Arctic fish and it's living where it's very cold, it would be very likely that it would have a higher cholesterol content in its cell membrane than other fish as well. And that might change it as far as eating the fish as well. That, you know, when you're talking about certain things having much higher cholesterol concentrations than other things, this could be one of the reasons why, because just simply their cell membranes are adapted to where they live. Okay, now the most important thing in the cell membrane, honestly, is the proteins, because all of the jobs that the cell does, it's going to rely on what can get in and out of the cell. And for the most part, most things can't pass through the cell membrane, not through the phospholipid part. That's what makes it selectively permeable. Really, what's getting selected are things that can pass through, for the most part, pass through the proteins. So the proteins are going to determine what the job of that particular cell is. They are the most important parts. They fold up so that the hydrophobic and hydrophilic portions align with the membrane. And here we are, our first one is transport. So transport is what basically takes molecules in and out. Most molecules cannot go through the phospholipid bilayer, particularly ions, particularly covalent things that are large, or polar covalent things that are large. So, proteins, sugars, and then all of your ions, and those are things that a cell needs. So obviously the proteins are going to be the key. A good example of this is in cystic fibrosis. We've used this as an example before. Remember, it goes back to the DNA having an error, which then causes a protein to be made wrong. It then folds up incorrectly, and in this case, this is a protein that controls the passage of chlorine or chloride ions. And so if we look here, we see this is the normal channel protein folded up the proper way. Chloride ions can pass through. Here's the protein from a person with cystic fibrosis, and this particular protein is folded incorrectly because of a DNA mistake that then causes the wrong amino acid. So you get the wrong secondary and tertiary structures. And then when that happens, that person's not going to be able to transport the chloride ions, and because they can't transport the chloride ions, then you end up with this mucus buildup. So that's a really good example of the importance of protein membrane, or membrane proteins that keep the cell alive. A second function of proteins in the membrane is that they can behave as enzymes. We're actually going to do a whole chapter where we talk about enzymes. Enzymes speed up reactions, and so membrane proteins could function as enzymes that are speeding up reactions that are happening at the membrane. The third one is support fibers. Your book actually breaks this into two separate functions because, for one thing, you've got the fibers that are holding membrane shape, anchoring it to the cytoskeleton, remember microtubules and all of that. But you also have, in addition to that kind of support fiber, you've got the cell junction proteins as well. Remember desmosomes and tight junctions and gap junctions and all of that. That goes along also. Like I said, they separate it, but it's pretty much, to me, it's kind of the same function. It has to do with supporting and connecting. The next one is receptors. This is really important because these kind of proteins pick up messages from the outside and then cause a reaction to happen inside. Most hormones, unless they're steroids, because steroids can pass through the lipid bilayer, protein hormones can't. So what they're going to do is they're going to attach to a protein, sort of like somebody knocking on the door, and that stimulates you to get up and go answer the door, or to get up and do something. A fire alarm goes off and you then respond in some way. So that's actually how receptor proteins work. A hormone will attach to the receptor and it won't actually enter the cell. The receptor will send a signal inside to then cause a particular reaction to happen. The fifth thing, the last one, is glycoprotein. So glyco has to do with sugars. We're going to learn a new type of sugar now. We've learned, remember, polysaccharide, monosaccharide, disaccharide. In this case, these are called oligosaccharides. Oligo means a few, because when we've talked about polysaccharides, we're usually talking about like starch and glycogen, and these are like 100 sugars long, really, really big, bulky things. Oligosaccharides are just like a little short chain, but these are really important because these are ID markers. And that kind of a protein, for example, determines your blood type. If you have a marker that's called A, your blood type is A. You have this actual marker on your red blood cells. If you receive the wrong blood type, that marker will attach to basically the incorrect blood that's given to you. You make antibodies. It causes this whole clumping reaction. It's also the reason why you couldn't just donate a kidney to anybody, because these surface markers identify your tissues as belonging to you. They're going to be more similar in family members, but at the same time, that doesn't necessarily guarantee you could donate a kidney to your sister or anything like that. Another thing that'll happen with these glycoproteins, they can be attached to things like in your immune response. You have certain white blood cells that will actually tag bacteria with glycoproteins that basically mark them, like put little bullseyes on them, sort of, so that the other types of white blood cells will then turn around and attack them. So ID markers can identify something as belonging, can also identify something as not belonging inside of the cell. Okay, and this is just a diagram. We went through all of these: transporting stuff, enzymes, attachment, glycoproteins, joining cells, and then receptors. Finally, to finish this up, what can and can't pass through the membrane. So I mentioned this before, lipid-soluble things can go right through. Also small things like carbon dioxide, oxygen, and water. Now water's polar, so you would say, well, I don't understand why water can pass, because you said earlier that polar things shouldn't go through. Lipids, water, they would reject each other. But water, there's actually little channel proteins called aquaporins, and water flows freely through those. So carbon dioxide, oxygen, water, and then lipid-soluble molecules are easily passed through the membrane. That's why toxins that are lipid-soluble tend to get stored in cell membranes, and you have a harder time getting rid of them. Things that can't pass would include large polar molecules, like sugar, for example, And then ions, hydrogen ions, sodium ions, chloride, magnesium, calcium, and these are all ions that cells need for certain jobs. So this would be really important, that you would need some kind of proteins to carry these things across if the cell was going to need them.